Hello and welcome to Dove Biology APES Lessons to Go. In this video we'll be exploring population analysis and conservation. The knowledge of species abundance and distribution is an essential part of conservation biology. By knowing how many individuals exist in populations of endangered or threatened species, we can establish an appropriate conservation plan. Additionally, for those species which are harvested, like the white-tailed deer, we can establish a management plan so that we know how many a hunter can remove from the population during each hunting season. Now, the results of population analysis can be fed into regional, national, and global databases for analysis to determine the effectiveness of conservation and management solutions and to provide an early warning of problems. For example, if we examine this graph that illustrates the population data for the house sparrow in England from 1976 to 2003, we can see that the population is in decline. By analyzing this data and relating it to other data, like for example the introduction of the house cat, we might be able to establish the reasonings behind the decline of this population and come up with a reasonable plan for its recovery. Now, as we collect measurements of population, there are two different measurements that we might obtain, either an absolute density or a relative density. The absolute density of a population is the actual number of individuals per unit area or per volume of water. This is really important for conservation and management of populations because if we know exactly how many individuals that are out there, we can establish an appropriate conservation plan. The relative density of a population is simply a comparison of the number of individuals between two different areas. Now, unfortunately, this only provides an index of abundance and is not very useful in establishing a conservation plan. Now, one way to determine absolute density is through total counts. A total count is achieved through the direct counting of all the individuals in a population, much like we do with the U.S. population census. We could go into a forest and count the number of trees, or if we have a breeding colony of organisms like these harbor seals, we can take a picture and then count that population later. In general, though, total counts are only possible for very few animals because typically animals are always moving around and it would be very easy for you to double count the same individual. Another way to determine absolute density is through sampling. With sampling, you use the count of a small proportion of the population to estimate the total population. For example, instead of counting all of these ants, we only count a few of those and use that as a proportionate example of the total population and extrapolate out to figure out the total population. Now, in general, there are two types of sampling techniques using quadrats and the capture-recapture method. With quadrats, what we do is we count all of the individuals in several small, typically rectangular plots, which we call quadrats. Then we extrapolate the average count to the whole area. For example, if this particular island was 250 meters squared in size, and each one of our little quadrats were 10 meters squared, we could count up all the individuals in each of the quadrats and then get our average. So we've got an average of about 1.4 individuals for every 10 meters squared. So how would that extrapolate out to our total island? So we cross multiply and divide, and we will determine that we will have approximately 35 individuals, which it comes pretty close to the actual number that is there. Now, there are certain requirements for quadrat sampling. For example, the population in each quadrat must be determined exactly. Since this is supposed to be a representative sample, we need to make sure that we count all of the individuals in that quadrat. We need to make sure we know the exact area of the quadrat, since this is the relative area that we're going to scale up to match the whole area. The quadrats themselves must be representative of the area, and typically this can be achieved by random sampling, because inevitably some of the quadrats may have few individuals, while others may have an overrepresentation depending upon the distribution of that particular species. Quadrat sampling is excellent for plants and animals that don't move around a lot. But for animals that move a lot, we need another technique. And in this case, we'll use the capture-recapture method. With capture-recapture, you're going to have a portion of a population that will be captured, marked, and then released. The mark could be a tattoo or a radio tag. Later, another portion of that population will be recaptured, and the number of marked individuals within that sample will be counted. Since the number of marked individuals with the second sample should be proportional to the number of marked individuals in the whole population, we can estimate the total population size by dividing the number of marked individuals by the proportion of marked individuals that we got in our second sample. Now, the time intervals between the two samples must be short, because this method assumes a closed population, with no recruitment of new individuals into the populations between time 1 and time 2. And it also assumes that we're not going to have any major losses of our marked individuals as a result of death. Now, to determine relative density, there's a couple of different strategies. We can observe the number of artifacts that were left behind by that organism. In some cases, the pupal cases of emerging insects or fecal pellets of different animals. If we find a lot of feces, we probably can relatively say there's a lot of individuals in that population. We can put questionnaires out to sportsmen or trappers so that we can kind of get an idea of how many fish or how many fur-bearing animals that they're capturing, because if they're having successful seasons, that typically might tell us that, relatively speaking, there's pretty good populations of those individual organisms. We might put bait out and see how quickly that bait gets eaten. If it's slowly eaten, then maybe there's few organisms that are attracted to that bait. If it's eaten really quickly, maybe there's a lot of individuals. We might look at ground cover, for example, with forest. We can estimate the average size and then how many trees that we can kind of see, and kind of extrapolate out the percent ground cover to the population of, say, for instance, trees. We might do some roadside counts so that as we drive along, we count the number of birds across a certain distance and then extrapolate that out to figure out the relative density. When it comes to conservation and management of populations, population analysis is key. The better we know the population standing of our various organisms, we're able to put together solid plans for conservation and management so that we can sustain biodiversity well into the future.